Hi, my name is Alexa Alvarez, and today we are working on Lab 8, and Lab 8 consists of carbohydrate fermentation. And this is my card. And so I kind of have preset some of the, set up some of the experiment just for, I want dead airspace, I guess. So we have the fennel red broths. So we have 6 of them. So each vial was supposed to be labeled. So 2 vials we're gonna be working with glucose. The other 2 we're gonna work with fructose, and the last 2 we work with mannitol. And what we will end up doing is we are going to add, and then also we have our actual sugars here, and we are going to be adding half of this. So for example, this is glucose, so half of the glucose is going to go into the glucose vial, and the rest is going to go in the second glucose vial. Same thing with the fructose and the mannitol. Then we have had these sitting, I've had these sitting in alcohol for a while already, just to sterilize. So I have both of the pipettes, and then we have, I'm sure I have 6. Yeah, 6 Durham tubes. What we will ultimately end up doing with these is we're gonna flip the little tubes into one of these, and we are going to, I'm sorry, no, we are gonna, first we have to add the sugars into each of the vial, and then we have to sterilize. We are also working again with our yeast and epidermidis broth solutions, or cultures that we created. So we are going to get started, and hopefully this works. I believe these are kind of at room temperature already. So okay, so we are gonna sterilize these. Since we're gonna start with, we'll just start, the instructions start with glucose, so we're just gonna start with that. So I have 2 glucose vials here with the fennel red solution. So we are to start, and then we have our test tube rack. It's the same thing that we've done throughout the course, so we have to sterilize the opening of the tube. It's a little bit of a draft in here. I have the air going. Just make sure both of them are glucose. come out. Okay, so we have that. And then we have to sterilize these again. Close, and we have to shake just to dissolve the glucose in here. Okay. Same thing with the second vial. Sterilize it, close it, and shake it. Okay, so now we're done with glucose. So we are going to work with fructose. So same thing, we're gonna open these up, sterilize them, add fructose, and then sterilize them again, close them, shake them, and let them hang out. Make sure everything gets dissolved. Okay. Good old fructose. And again, we're going to sterilize these, seal them, and shake them. Fructose crystals are a little bit larger, so I'm assuming these will probably take a while to dissolve. Not too bad. And last, we're going to work with mannitol. Here. A little bit more. This is a lot finer of a powder. Okay. So now that we have that done, we have to take out our Durham tubes out of the alcohol, and we have to let them dry. alcohol inside each of the tubes. So. So we're gonna let these dry for a while and then we will be back. All right, so I think these are dry. So we are going to add, place 1 dirham tube in each of the fennel red tubes. And then we have to make sure that there isn't any air trapped inside of the tubes. I think there may be a little bubble in there, but we'll work with that in a little bit. Okay. Okay, so now we're gonna work with our good old staff and yeast. So I'm sure these are not gonna smell very good. So these have been drying. I took them out when I took the dirham tubes out, so hopefully, yeah, they look like they're drying pretty well. Okay, so we have to add 4 drops of the cultures in each of the tubes that are corresponding with each culture. So what I am going to do is I'm gonna make sure, pretty much I'm gonna set up 2 rows. So the front row so far seems to be the epidermidis, and then the back row it looks like they're all yeast. So that I don't confuse them, I'm gonna keep them front and back, like separate, just so that I know what row I'm working with and what culture. So we're gonna add, it says 4 drops, and then obviously we have to sterilize both tubes, and then we have to, after this, they're gonna sit for about 12 hours, and we're gonna see if we notice any fermentation or reaction. Okay. Okay, so I have everything sterilized. So now we're gonna add 4 drops into each of the staff vials. Okay, that's done. Okay. We're gonna flip. These aren't very good test tube Rex. That's okay. Okay. Now we have yeast. Four drops of this. Okay. So it is, let's see, it is about 11:20 in the morning. So we have a late day today. So we have to incubate these for 12 hours. And then we have to see if we notice any color changes or anything after the first 12 hours. And then if I remember correctly, we're gonna look and see if there's like a yellow orange color or if there's no color, and that'll tell us if there's reaction happening or if the solutions are fermenting. And if we don't see any changes after the first 12 hours, we're gonna incubate for another 6 hours. So hopefully we see some reactions. And I guess we'll be back later tonight and see how everything looks. Hello. So we are on our second part of our experiment, which after incubation, we have taken it out of the closet area where I keep everything, and I have results. So the first, well, I guess we'll go over results later, but I just wanted to show kind of the outcome of what we got between all 6 broths, and I think that is all we have for now. So this will conclude the experiment video, and up next we'll do results. Thank you so much.